Chapter 1 Introduction. Background of the study. Milk is an important food for infants and growing children. It is very expensive and extremely perishable, and this has led to the development of alternative sources of milk from plant materials. Canarium ovatum, or locally known as a Pili, is a species that is native to the Southeast Asia, Papua New Guinea, and Northern Australia. The pili nut has been claimed to be the king of nuts due to its nutritional value. Lactic acid bacteria is an acid-tolerant bacteria which helps in the digestion of lactose and has been used in probiotic drinks. This study aims to determine the aid provided of lactic acid bacteria fermentation in terms of overall acceptability and nutritional content of the pili nut milk. Significance of the study. Milk is an important source of calcium for keeping our bones strong and healthy. The results of the study may aid people who have digestion problems since lactic acid bacteria helps in producing protective mucus and reducing instances of bloating. Through this study, several farmers will be given more opportunities in growing local goods such as pili nuts. This study will also be of help to future researchers in the same field in providing data with the information gathered during the course of the study. Scope and limitations. In the microbiological assessment of the milk, the types and the total count of lactic acid bacteria found in the milk shall be given more emphasis. As for the nutritional assessment of the milk, protein and fat content shall also be evaluated. The study will be limited exclusively to the usage of pili nut as plant material for the alternative source of milk, and lactic acid bacteria for fermentation.
Review of related literature. Milk has been recognized as an important food for infants and growing children, but in developing countries, the cost of dairy milk and their products is prohibitive, and this has led to the development of alternative source of milk from plant materials. An inexpensive milk substitute could be extracted from locally available plant foods like legumes. Milk is extremely perishable, and many means have been developed to preserve it. The earliest one of which has been used for many thousands of years is fermentation. Milk can be fermented by inoculating fresh milk with the appropriate bacteria and keeping it at a temperature which favors bacterial growth. As the bacteria grow, they convert milk sugar, or lactose, into lactic acid. You can detect its presence by the tart or sour taste. Pili belongs to the Canarium genera of the Berceraceae family. It is believed that there were 53 species of the Canarium genus that used to thrive in the Philippines. But its number has been reduced to 9. At present, the two most commercially important species are the Canarium luzonicum and Canarium ovatum. The latter one would be used for arsenic. Its kernel is rich in magnesium, phosphorus, potassium, calcium, ascorbic acid, riboflavin, zinc, copper, iron, sodium, thiamine or vitamin B1, and niacin or vitamin B3. The kernel can be eaten raw or roasted. Fermentation. Fermentation is a means by which cells growing anaerobically can still generate a little ATP or adenosine triphosphate. During the breakdown of sugar, known as glycolysis, excess hydrogen atoms are generated and must be deposited somewhere. In lactic acid bacteria, they dump excess hydrogen atoms on pyruvic acid, the end product of glucose. This turns pyruvic acid into lactic acid. Species of lactic acid bacteria, or lab for short, represent as potential microorganisms and have been widely applied in food fermentation worldwide. Milk fermentation relies heavily on the activity of lab, where transformation of milk to good quality of fermented milk products is made possible. Lab have a role in milk fermentation to produce acid, which is important as preservative agents and generating flavor of the products. They also produce exopolysaccharides, which are essential as texture formation. Considering the existing reports on several health-promoting properties, as well as generally recognized as safe status of lab, they can be widely used in the developing of new fermented milk products. Probiotic products are usually marketed in the form of fermented milks and yogurts. However, with an increase in the consumer vegetarianism throughout the developed countries, there is also a demand for vegetarian probiotic products. Meanwhile, lactose intolerance consists of the absence of lactase production, which is responsible for hydrolyzing the lactose present in dairy products. According to the National Institute of Kidney Disease and Diabetes, affiliated to the American Institute of Health, about 75% of the world population is lactose intolerant. As for related studies, a research entitled Microbiological and Nutritional Assessment of Starter-Developed Fermented Tiger Nut Milk, made by the researchers Sherifa Monilola Wakil, Oluwatobi Tolu Ayenuro, and Kubrat Abiola Oyinlola, they have produced an excellent and acceptable FTM, or fermented tiger nut milk, which brought positive results.
Chapter 3, Methodology. Making the nut milk. The researchers will make 4 batches of the nut milk, blending together 1.5 cup of skinless pili nuts, 2 cups of water, and 2 tablespoons of sugar. Afterwards, they will strain the mixture using a cheesecloth or muslin cloth, leaving behind the liquid, which is now the nut milk. They will then boil the milk for 5 minutes and, while still hot, put it in a sterilized bottle and leave behind a 2-inch headspace. Then the milk will be boiled for 20 minutes in a water bath. Afterwards, it will be cooled for 5 minutes in room temperature and then cooled with lukewarm tap water. Finally, the milk will be stored in a refrigerator. For lactic acid fermentation, Yakult will be used as a starter culture. Different concentrations of Yakult will be used. Fermentation will be done in a room with 25 degrees Celsius room temperature and will be done in 3 to 4 hours. For the Microbiological and Nutritional Assessment, the Food and Nutrition Research Institute in DOST Taguig will be of assistance. They will help in determining the lactic acid bacteria count and the protein and fat content inside the nut milk. And finally, texture, appearance, aroma, and overall acceptability of taste will be evaluated for the sensory evaluation. This will be done with the help of a food scientist from DOST and also by the 3 researchers.